Hi, and let's continue our discussion on sequence spaces and fitness landscapes. So, what we had done last time was introduce the fact that given a DNA sequence of length L, there are in all a 4 to the power L number of sequences which have the same length L, but different nucleotide sequence. Similarly, if we are talking of a protein which has 100 amino acids in it, there are 20 to the power 100 polypeptide chains, each one of them equal to length 100, and length here refers to the number of amino acids which go towards forming that polypeptide chain, but the constituents of that 100 amino acid chain are different in each one of the 20 to the power 100 amino acid chains. And we had stopped our discussion by representing, we had just started towards representing these sequences 4 to the power L sequences or 20 to the power L sequences of amino acids in a graphical sense. So, let us continue our discussion towards that, and we will come to the definition of what is referred to as the fitness landscape. So, we will start our discussion by the example of DNA. So, we have a DNA sequence and its length is L, and we have already done that if we were to define a sequence of length L, there are 4 to the power L number of sequences. Now, what we are going to do with these 4 to the power L sequences is think of this as a 2-dimensional plane and do a graphical representation of these 4 to the power L sequences, where each node represents one of the 4 to the power L sequences that we are talking about. Okay. So, let us say node number 1 represents that sequence of length L which is all A's. This is node number 2, this is that sequence which is all A's, but the last nucleotide is T, and so on and so forth. We keep doing this till we populate this plane with these nodes, and the total number of nodes in this plane will obviously be equal to 4 to the power L, because every distinct genotype or the DNA sequence that we are talking about will be represented as a node here. So, at the end of the whole exercise, we will have 4 to the power L nodes on this plane of paper. Let us draw some of them. Let us say this is the 4 to the power L nodes that we are interested in, and this is the last one, which is node number 4 to the power L, this is node number I, and so on and so forth. So, we have populated this plane with these nodes. Now, what we are going to do is we are going to join node I and J. Let me write that down on a separate sheet of paper. The rules are, what we want to define a structure, and we want to define the rules about how we are going to go towards defining that structure. The first exercise that we have to do towards explaining fitness landscape, which is a structure we are interested in, is represent all possible DNA sequences. In this case, where we are talking of DNA, in the case of proteins, it will be a similar approach towards defining that fitness landscape. So, in DNA sequences, first we represent all possible DNA sequences, and each sequence is represented by a single node. So, we do that, and we have all 4 to the power L nodes here. Next, what we are going to do is join node I and node J. So, this represents, node I represents the DNA sequence corresponding to that particular node, because remember, each node corresponds to a distinct DNA sequence which is of length L. Now, we are going to join node I and node J if they differ at one position only. Which implies that at L minus 1 places the two sequences I and j are identical. Let me make that clear again. Let us say if L is equal to 4, that means I am only interested in understanding sequences of length 4, that is all I am interested in. Then the total number of sequences that I am going to end up with is 4 to the power 4, which is 16 into 16, which is 256. So, I will have 256 nodes drawn on the plane of my paper. Now, each of these nodes represents a particular sequence, and I join two nodes if they differ at one nucleotide position only and the remaining 3 are the same. For instance, if this node refers to A T and this one refers to A G, that means the first 3 nucleotide positions here and the first 3 here are the same. These two are distinct. So, these two sequences, sequence I and sequence J, only differ at one nucleotide position, which is one position only, and at the remaining L minus 1, which is 3 here, the two sequences are identical. And if that is the case, then these two sequences are joined by a node. Let me take another example. If you have a sequence called AATA, which is represented by this node. Now, what we want to do here is say that the question is whether these two nodes should be connected or not. In one sense, these are identical in the sense that they have 3 A's and 1 T. This one also has 3 A's and 1 T, but these will not be connected because we are interested in the specific order of the nucleotides that we are talking about in a particular sequence here. Of the two sequences that we are looking at right now, they are only identical at 2 nucleotide positions and the remaining 2 nucleotide positions are different in them. If we again look at them carefully, these two are identical at the first 2 positions AA and AA. After that, the third, this sequence has an A, this sequence has a T, that is a difference. This sequence has a T at the 4th position, this one has a A at the 4th position. Hence, these two differ at not 1, not 0, but 2 positions, and they are only identical at L minus 2 places, which is 2. Hence, we will not join these 2 nodes with a edge here. You could also have differences in the middle, which is you could have AATT. This is the node that corresponds to AATT. And now, if we are talking about these two sequences, they are identical at 1st, 2nd and 4th position. However, they differ at the 3rd position, which again satisfies this rule that they only differ at one position only, the other 3 are identical, and hence these two nodes will be connected by an edge, and so on and so forth. You keep building that for all 4 to the power L nodes that you have in the original scenario that we started with. And if L is the genome of an organism, we are talking of very large number of nodes. Right? So, again we connect 2 nodes if they only differ by one nucleotide position. What is the significance of connecting them if they only differ by one nucleotide position? We want to do that because via this linkage, this gives us an example or an idea about how a sequence might jump from one particular sequence to another particular sequence. For instance, if I have a population such that its genotype is this one, AATT, all n individuals belong to this particular genotype. Now, what might happen is that one of these individuals might acquire a mutation, and that mutation is going to be a single nucleotide, which is called a point mutation, and during that mutation this T might get changed to an A, which means the mutant individual has jumped from this sequence to this sequence on my graph. Now, depending on what is the growth rate, whether this beneficial helped the individual or not, let us imagine that the beneficial that had happened was beneficial in nature, the mutation that happened was beneficial in nature and that resulted in this mutant growing faster than the rest of the population. Then that mutant starts, we go back to the selection and the mutation rules that we have just studied, and we can easily see that should that mutation have been beneficial in nature, the mutant that has arisen at this point will start eliminating the rest of the population and eventually the population moves to this point. And now again that same story repeats itself. Suppose we have a population of n size n occupying this genotypic space, all n have the same genotype, and now should a mutation happen where this A gets changed to T, we have one individual which has come back to the original position, original genotype which was there several generations back, but now we know that the fitness of this genotype is more than fitness of this genotype, and hence that mutant individual is no longer going to be able to outcompete the parent genotype from which it mutated from, and hence this genotype survives. But what happens if now this T changes to a G? Now what that means is we have a mutant which is of this genotype, and if this genotype grows faster than this genotype, then the mutant which has come at this place is going to be able to outcompete everybody here, and then the entire population moves towards this node. So you have movement of populations taking place on this graph where each sequence is represented by a node, and nodes are connected if you can go from one sequence to another via a single point mutation. In our case, node this and this node are not connected because you cannot have a single point mutation occur to this sequence and automatically reach this sequence. You need to go through an intermediate sequence and hence approach that one. Right? That is the first thing that we want to understand while we go towards understanding fitness landscapes. And the concept is similar for proteins, and now for proteins we are again going to look at the example that we had started last time. We have a protein of length 100, so the total number of nodes which is going to happen is 20 to the power 100, where 100 is the length of polypeptide chain. And each one of these, this is number of sequences, and each one of these sequences is going to be represented by a node. This is sequence A, this is sequence B, and so on and so forth, going all the way up to sequence number 20 to the power 100. And the rule for connecting nodes remains the same as the one in DNA, that when we are talking of protein sequence spaces, two nodes are connected if the sequences that they represent only differ at one amino acid at a particular position, only differ at one amino acid location. If the length of this polypeptide chain is L, then the remaining L minus 1 positions are identical in the two sequences. The point of this whole exercise being that to try and understand that whether you are dealing with DNA sequences or polypeptide chains, what you can represent them as these nodes and edges, where each node represents a sequence and two nodes are connected by an edge, if via a single mutation in nucleotide or amino acid you can jump from one node to the other. So, we have this. So, irrespective of whether we are talking DNA or proteins, we have this connected edges and nodes diagram now. So, the first question that I would like you to think about is that how many neighbors does a node have? And let us first think in terms of DNA sequences. So, the question is that we have this nodes and edges diagram, we have a bunch of nodes 4 to the power L nodes, and not all nodes are going to be connected to every other node. We have 4 to the power L nodes, and any node is going to be connected to a subset of these 4 to the power L nodes. A node is not going to be connected to every other node in this graph, but only going to be connected to a subset of them. And the question is, how many neighbors does a node have, or how many nodes does a particular node have as neighbors? And remember the rules that I and J are connected if they only differ at one nucleotide position. So, what we are interested in is that out of the 4 to the power L sequences, how many sequences can a particular sequence go to via acquisition of just one nucleotide position. So, if I represent a node, I represent a particular DNA sequence of length L. Should a mutation happen, a single point mutation happen in me, how many of the other 4 to the power L sequences can I generate, and hence I will be connected to each one of these other sequences that I can turn myself into if I were to acquire one point mutation in my sequence. So, what I would like here is for you to pause the video for a few minutes, try and work this out by yourself. We will work through the answer after that. So, the way to think about this problem is that let us redraw our example. These are our 4 to the power L nodes. This is 4 to the power L nodes, and let us say this is the node that I am interested in, and I am interested in computing the number of neighbors of this particular node. How do I find that out? 4 to the power L comes from the fact that there are L number of sequences, L number of nucleotides in each of these sequences. In each sequence, right? And each sequence here is represented by a node. Now, if that is the case and I am only interested in talking about single point mutants, nodes are connected if they only differ from one another at one particular position and are identical at the other L minus 1 positions. So, if that is the case, that one difference, so we are interested in sequences. Let us give the node that we are interested in some name. So, this is node N. We are interested in sequences which are represented by nodes. So, nodes and sequences are going to be used interchangeably in this discussion. We are interested in sequences or nodes which differ from node N, the sequence corresponding to node N at one position only. those are the nodes that we are interested in. And of the total available nodes, we have 4 to the power L minus 1 available nodes. Because one of the nodes is the node n itself, so we subtract that. However, because L is such a large number, for all practical purposes, this is just equal to 4 to the power L. So, we are just going to keep talking as if the total number of available nodes is 4 to the power L and not 4 to the power L minus 1. So, that is what we are interested in. We are interested in all those sequences or nodes which differ from the sequence defined by node n at one position only and are identical at n minus 1 positions. So, how do we talk about this? That difference that we have to talk about. So, we are interested in sequences or nodes which differ from node n at one position only. That difference could be anywhere in the sequence. We have not specified that. That difference could be at the first position and nucleotide number 2 to L could be identical. That difference could be at the second position. The first nucleotide could be identical and nucleotide number 3 to nucleotide number L could be identical, and so on and so forth. So, that one difference could be anywhere in the sequence. And anywhere, by anywhere I mean the position number of the nucleotide. If that difference is happening at the first position, then my question is, how many sequences are there which differ from sequence or node n at first position only. So, the difference between this node and this node is one nucleotide position. But that one difference could be anywhere. That could be at position 1, that could be at position 2, going all the way up to position L, but it could only be at one position only. So, we are going to try and do this systematically and try and identify the number of nodes which are different from the sequence corresponding to node n at position 1 only. The remaining positions, position 2 to position L, the remaining positions sequence from nucleotide number 2 to nucleotide number L are identical. Okay. So, this is the question that we are interested in under these constraints. Take a 30-second pause from the video and try and answer this question. The way to think about this is, I have my node n and its sequence is of length L, and I am going to divide this into 2, where this is nucleotide number 1, the position of this nucleotide is 1, and this chain represents nucleotide 2 to L. If that is the case, then I am interested in how many neighbors does this node have which only differ at this nucleotide position and have the identical nucleotide sequence from nucleotide number 2 to L. And the answer to that would be 3. And the 3 will be because I am not free for the neighboring nodes. If the difference is at nucleotide position 1, the neighboring nodes do not have any choice when it comes to nucleotide position 2 to L. They have to take on the same sequence which node n that we are talking about has, because this node and this node only differ at one nucleotide position. However, they are free to take a different nucleotide at position 1. So, imagine that the nucleotide at position 1 in my node N was A, then one of the possible neighbors could be a T at the first position and the same identical nucleotide 2 to L in at the for the remaining sequence, or it could be a G or it could be a C. So, the number of neighbors that node N has, which differ at only the first position of the sequence, are equal to 3, and hence the total number of neighbors for node N will be number of neighbors which differ at position 1 plus number of neighbors which differ at position 2, which can be analyzed in an identical way as to what we have done just now, and going all the way up to number of nodes which differ at position L. And these numbers are just equal to, we have just seen that this number is equal to 3. Similarly, it can be shown that this number will also be equal to 3 and going all the way up to 3 for every position where the difference between node N and its neighbors exist. And these are L number of 3's added, so the answer becomes 3L. So, of the total 4 to the power L nodes, a node is connected to 3 to the power L nodes. As an exercise, you should try and do a similar exercise for a protein peptide change for a protein chain of for a protein of length L. Construct all possible polypeptide chains which have an equivalent length L, and then try and decipher that of the 20 to the power L nodes that you have in your graph, how many nodes is one particular node connected to via the same rules that we have been defining so far. We will continue this discussion in the next class onwards. Thank you.